To introduce today's speaker.
All right, I was told to stand in the middle here, or Stephen was going to get mad at me. So it is my great pleasure to be able to introduce Walter Jetz, professor at Yale, if you guys didn't gather that, in the Department of Ecology and Evolutionary Biology. And you know, I have a little blurb here from the website, but I'm just going to say that I know that Walter's a birder at heart, so he definitely fits in here. But I thought I'd start with a disclaimer, first of all, that so Walter's our lead PI on a genealogy of life grant that Rory is heading for the MVZ. So we're working on the terrestrial vert life, where we're going to be combining all terrestrial vertebrate phylogenies and traits together. It's a very exciting project. So it's been a pleasure working with Walter in his lab. But like I said, you know, I've taken a look at your entire publication list, so I can see that you are definitely a birder at heart. But I also noticed there were papers there that mixed in with badgers and aerial insects, and so there's definitely a much broader interest there to try to understand, you know, the global biodiversity essentially, and you know why species occurs where they do. So it's entirely appropriate and perhaps just a natural evolution that you're also the lead PI for the Map of Life project. So I don't know how many people have heard of the Map of Life, and I think maybe we'll be hearing some of that today, hopefully. But it's a fascinating and amazing project that brings together cutting-edge informatics and data in a very web-accessible and very user-friendly tool to map out species distributions and basically everything that we know. And it doesn't just cover birds; it covers all of life. So it's so that's actually, I think, where Walter is making a huge impact. But he has a number of other really exciting papers and impacts on our understanding of bird phylogeny in particular. So if folks are interested in that, they should definitely take a look at a number of nature papers, in particular the global bird phylogeny, which is also available on the website, which I think is
Birdtree.org.
Birdtree.org, okay. I was also asked about a fun fact about Walter. So I will say that before he went to Yale, he was also a professor at UC San Diego. And despite having been from Germany, I noticed you used your years at San Diego wisely, and I've heard that you've become a pretty good surfer. So I don't know, we'll hear about that. But so it's my pleasure to introduce him. And the title of his talk, Reconciling the Effects of Time, Area, Ecology Constraints, and Energy on Geographic Gradients and Species Richness. So, welcome.
Thank you, Michelle. Thanks for having me. And I think the main fun thing about the surfing part could be more like watching me now trying to get back into shape again. And it's a pretty bad wipeout, so definitely some lost time there at Yale. But anyway, so thanks for having me. And today I'm gonna sort of complement a little bit to what I talked about last Friday over in ESPM, where I spoke more about some of the policy relevance of our work, conservation relevance, especially on the spatial biodiversity front. But today I thought it'd be fun to follow up a little bit on what seems like at least a partial theme of the semester here set off by Paul Fine's talk a few weeks ago that I'm sure many of you were at, on spatial gradients in biodiversity. And I thought this could be an interesting way to follow up, connect to Paul's talk and sort of drill further into that theme. And it's been really fun, of course, collaborating on that with Paul, but we're coming at it actually from a somewhat complementary and fun, in a fun way, different perspectives. And so it's been really great working with him. And so let me now expand on that work a little bit and start out with a bit of a personal story, how I got into this whole business of studying birds, and of course since branched out, and in particular trying to understand the spatial variation in biodiversity, attributes of assemblages such as the species richness. So I would find myself as an undergrad running around in this field station in Comoe National Park in West Africa, and sort of walking around really and trying to understand how it is then that we see that number of species, of bird species, at that place that we see. Why do they have that sort of structure, that body size distribution? Why do they have the richness that we see? And I would sort of very much, a little bit like MacArthur set out in some of his writing, sort of run around and try and study the community very closely, right? Understand how species differentiate their niches, how it all really works, how it is then that these species coexist in that place. But as I did that, I increasingly during my PhD then started to appreciate that I can run around in that location as much as I want and study the niche partitioning, and I would still not be able to understand why it is then that we have that particular setup of species. And I would find myself from the first year of my PhD towards the last year increasingly sort of zooming out and starting to take a larger and larger perspective, starting to appreciate that we're sort of in a transitional biome here from the wetter forested parts of Africa to the drier Sahel parts, that you have species reaching in here from the forest biome, your species reaching in here from the Sahel biome, and you have really an overlap of different sets of species with different historical biogeographic backgrounds, if you will, come together. And it's really was an interesting experience during my PhD to find myself zooming out more and more, and in the end I found myself writing my PhD on the richness variation of all African birds, with the data that I would have collected in the field and myself only making up about a 0.2% or so. But that was just a necessity, because I felt like I gotta zoom out, and so I had to collaborate and work with other people to really get to that large scale. Now, that then set off in my own work, and many other people have been doing some really excellent work trying to describe these richness patterns at increasingly large spatial scales, and we've been able to do that in a really good way, relating that to then correlates such as productivity, and doing, I think, a really interesting advance in getting a grips with these patterns and their correlates. But what was always frustrating here, and it's sort of already charted out in this paper, is that whatever you're looking at in terms of a correlate of grid cell richness, so these would always be like gridded, 100-kilometer gridded patterns, because that's the only reliable grain or spatial resolution for that sort of data available, that when we look at these gridded patterns, we're really kind of conflating ecological and evolutionary processes, and most of all, we're also double counting species. So the majority of this pattern is really driven by just a quarter of species. It's the wide-ranging species that really drive the overall richness pattern. So you're really kind of conflating evolutionary and ecological interpretations here, because you're weighting some species much more than others. You're not accounting for the importance of some of these narrow-ranged species that barely actually contribute to this overall richness pattern. So that's a real limitation to just analyzing these gridded patterns. In other work, then we got to appreciate more and more that also just analyzing these data sets at one spatial grain at 100 kilometers is really very limiting, in that as we actually take finer scale data from many places and then take a different spatial grain view, so we would like analyze these data sets at larger and larger spatial grains, we would find that the importance of environmental correlates changes expectedly, right? But it's never been quite appreciated. So we would always analyze data at this 100-kilometer grain or so. That is what the data usually allows us doing, these expert range maps, these blob maps, right? When we go to finer grain data, such as your national park checklists or whatnot, then we actually find a very different effect of, for example, habitat heterogeneity. And so do we, as we zoom further out to and look at a coarser and coarser picture. Now, let's talk more about net primary productivity in particular. So there's been all sorts of theory and good empirical work trying to address the effect of net primary productivity, or energy availability, or energy flux through the system, or food availability for these consumers on overall richness and coexistence and on richness. And it's really important to appreciate that this effect actually has a scale dependence. And it's not surprising because I think different processes are behind this effect at different spatial scales. And that brings us to this classical view at processes and their respective scales from Schmieder and Wilson that reminds us how as we go from local assemblages to the continental scale, of course, very different processes apply and have a different weight. As we go out, we look at the continental scale, we increasingly need to account deeper time effects, historical processes. Broad-scale environmental constraints may come in more at this sort of scale, and it's only really at the very fine scale of interaction at local assemblages that some of the typical community ecological biotic effects come in. So we really need to appreciate this scale dependence, and much of the work on trying to get to grips with environmental predictors of richness and the causes behind that, I feel we haven't quite sufficiently taken that into account, because of course it's difficult, and we are really limited by also our empirical evidence that tends to be very spatially restricted here and very coarse grain out there, and I'll come back to that later. We've also been restricted by the fact that people tend to sit in different disciplines, go to different meetings, and I feel though that if we want to get to understand factors such as energy availability and how and why they affect variation in richness, we do need to think about how energy effects on some set of processes such as energy availability, as diversification, may play out over different spatial scales and only come out at those sorts of scales when we analyze our data than factors affecting coexistence. Yeah. So energy availability may have a very clear effect on coexistence and thereby drive richness, but we would only discover that if we look at certain spatial scales. So to summarize that introduction, I feel that the drivers underpinning diversity, such as speciation and extinction, so some of the coarser grain stuff I mentioned earlier, would operate at scales much larger than that of ecological communities or these 100-kilometer grid cells. Yeah? Deeper time evolutionary processes are separate from recent ecological sorting and facilitators of coexistence that would operate at these finer spatial scales. And if we really want to get to an integrative understanding of the determinants of spatial variation and richness, we need to adopt a sort of hierarchical cross-scale framework. And that's what I'm going to talk about for much of my talk, this attempt to in a simple hierarchical way bring together and initially separate, but then bring together the drivers of diversification with those of coexistence or sorting at finer scale communities. And what I'm going to talk about are essentially three papers. One is on the global gradient, so this initial cross-scale framework, and Paul Finer already talked about it a few weeks ago. And so I think I can go through that relatively quickly, just to remind ourselves of this hierarchical view. I will then briefly touch on an attempt to bring ecological constraints into the picture. And then finally take a new sort of evolutionary, macroevolutionary view on how we could understand energetic constraints on species richnesses. And this has been worked with collaborators Paul Fine and Jonathan Bellmaker, former postdoc who is now in Tel Aviv as a faculty, and then Alex Pigot and Joe DeBias over in England. So Paul covered already last time this idea of bioregions as evolutionary arenas, and that is this coarser scale that I'm referring to here, that I talked about in my earlier slide, right? So this continental scale. How do we actually deal with spatial units at that scale? Well, bioregions offer a really good way of doing that. We're all aware and well appreciate the niche conservatism that there is in how clades tend to diversify into certain environmental conditions and then kind of stick with them. So even though as some of these bioregions may have shrunk or even moved over time and then expanded, we'd assume that many of the clades that involved in these regions, these bioregions, bioclimatically defined regions, would have stuck with them, and we can therefore take them as evolutionary arenas, if you will, in which we can understand diversification processes and their potential drivers over large temporal and spatial scales. And we find that indeed, when we delineate the world like that, for vertebrates, they indeed tend to characterize these relatively closed assemblages, if you will. Yeah. Now, let's look at a broad-scale productivity gradient. So this is usually how one would have analyzed variation in richness, a gridded pattern where these would be species ranges, and then you would have colder, low productivity, and then warmer, higher productivity places, and you find that species sort of extend over several of them, and then you would analyze the resulting richness pattern of grid cell richness against environmental gradient, say, productivity. Now, what we try to now appreciate with this bioregional approach is that these are not the same regions that these two different environmental conditions exist. It's region A here would be your temperate forests, and region B, for example, would be your tropical forests. And those regions really have very different biogeographic evolutionary histories. So we have in one region perhaps the case that it's arisen relatively recently, and there's been very little time for diversification. In another region, say the tropics, would have been around for a long time. If you take the gridded perspective, we are not able to take that into account at all. We're ignoring the fact that we're looking at very different regions and different histories. So perhaps we should first start out understand where that regional richness variation comes from, and then from there go to understanding how it is then that within a region along an environmental gradient species coexist. Okay, so that's kind of this hierarchical idea. Let's get to understand this here first before we understand that sort of pattern. And to get to an understanding of regional richness, we really want to understand the drivers of species accumulation. And I did say that productivity, of course, would come in here in terms of niche availability and coexistence, but it would also potentially come in over here. So these are all factors that have been suggested as being important for affecting the variation in regional species richness. Time, how long a region has been around, or a bioregion, so habitat, such as tropical forests in South America, the area, the extent of that region, then the overall productivity. Is it a highly productive region? Is it a low productivity region? Or the temperature? Is it a region where any fluxes happen at a very, very rapid, or interactions and fluxes happen in a very rapid way, or not? So here, the potential factors that have been suggested, so more time for speciation, for example, for time, larger areas would allow greater opportunities for spatial isolation, decreased extinction risk. Here, higher productivity regions would allow greater population sizes, less extinction risk, and then here in higher temperature regions, we could argue that there could be faster rates of evolution, and everything would just happen at a bit of a more rapid way, especially for ectotherms. Now, these two could be combined in a time-integrated area variable. This is something that Paul Fine pioneered. And finally, we could bring in another variable here that I'm going to cover briefly, this idea that trait diversity, capturing ecological constraints, could be an additional predictor. So low levels of ecological divergence among a region's lineages may impede the diversification of these lineages. I'll talk a little more about that in a moment. And all of these things would be affecting diversification rates. Now, we had a chance to take a first global look at the variation and diversification rates recently by putting together a full phylogeny for the birds of the world and mapping out these species level estimates of diversification rates that lead to a species. And we find that there is great heterogeneity in the tree as to where the faster diversifying or the more slowly diversifying clades are, and there is equal heterogeneity in the globe. But there isn't actually a very straightforward pattern in that the tropics would be the places that harbor species or clades that diversify more rapidly. Instead, we didn't find a signal of latitude at all on diversification rates. And that's quite a big surprise, actually, in many ways, in that it's always been argued as one of the key reasons why the tropics would harbor more species, is that they have just a faster rate of diversification, and over the same time leading to a greater number of species. Now here we are looking at the same pattern, but in this bioregion view, and here the richness pattern, here the diversification rate pattern for birds, and you see that there isn't much of a concordance between those two things. And in mammals, we found the same situation. So we're looking at the average diversification rate of clades occupying these bioregions. So in this follow-up study, we tried to address an additional predictor. We tried to address this idea that niche partitioning among species presents a key ecological limitation on coexistence, so that depressed within region opportunities for differentiation or low levels of evolutionary divergence in ecological traits among a region's lineages could impede within region survival of these lineages and therefore impose constraints on diversification. So the more similar clades are ecologically, the harder it is for new ones, for new species to actually arise and then survive. So we're asking the question, are differences in ecological divergence a dominant contributor to among regional variation in species? And for that, this is kind of one of the initial steps that also then led to the BirdLife project. We compiled a large trait database that we put online, so you can download it if you want, for birds that characterize their diets and characterize their core feeding heights or foraging heights, body size of course, and that allowed us to then do a phenetic tree of differentiation of all the species. So we could calculate as a first order estimate whether species are very similar or very dissimilar, and we were able to characterize regional assemblages with regard to that. So we were able to calculate median trait diversity, and of course in the species rich regions you'd have a much greater trait diversity than in species poor regions. The more species you add to this, you can think of it also as a sort of large hypervolume of different traits, right, coming together. The more species, of course, you'd expect you're occupying a larger and larger volume as you're adding in all sorts of combinations of body size and insectivory or different diet combinations. Now, we, in a second step then, felt like we wanted to control for species richness. We really wanted to understand the per species trait diversity that you find in a region. And a really interesting picture emerges. You find that in birds, here for example in South America, where you have a few clades such as the ovenbirds, tyrannid flycatchers, having brought about lots of species that actually are on the face looking very, very similar, tend to look like they're doing very, very similar things, occupying similar foraging heights, that you've been adding on species that actually did look very, very similar, and that on the overall analysis then you find that there is great on average species trait similarity, or there is very little trait diversity once accounted for species richness. In mammals we found a kind of a similar pattern. So we can put this together and then just compare these bioregions in how much median diversification rate or this median trait divergence may explain some of the among region variation in species richness. And as we do that, we find what we found in Jetz and Fine 2012 confirmed that time area, again and again, even as you add in other variables, comes out as the strongest predictor. So how long a region has been around and how large it has been over its time of existence is a key predictor, the core predictor, even in a sort of structural equation setting of analysis. As you account for all these cross-collinearities, it comes out as the main predictor of the variation in species richness among regions. Diversification rate explains very, very little at all, and this niche diversity metric, so how much species in a region are similar or not, so that idea that could be constraining in some way or facilitating greater richness is not supported by this analysis. So we find this effect here confirmed. In this setting, we found not a strong effect of productivity, and I'll come back to that a little bit later. So after we've now developed this model of regional richness, so our best attempt right now to predict variation in species richness, we can then go from there and see, okay, once we've explained this, what's left over to explain by these gridded analyses? So here, now, is this prediction for birds, mammals, and amphibians across the different bioregions of the world of the grid cell richness, and we find that we're able to do a very, very good job just with this regional model. So there's nothing else, and we're not using any grid cell level, 100-kilometer level predictors. We're just taking our region, time integrated area, and productivity thrown into the mix here, and temperature to predict variation in richness among these 36 regions or so what it was. And we find that that model alone explains grid cell, 100-kilometer grid cell variation in richness in a really, really strong way. And when we then add in within region variation in productivity, so the proportional regional productivity, so whether a cell within a region is a low productivity cell or a high productivity cell, then we're able to push that even further. So it's a very simple hierarchical model that can do an immensely, at least from a statistical perspective, good job in predicting species richness. Now, let me talk about the third study here, and that is one where we now try to get to that grid cell level, to that coexistence level. And here, let me start out with this productivity map, and then that assemblage richness map again for birds. And let's look at a kind of a new way, a different way of trying to get, from an evolutionary perspective, at what is it then that facilitates coexistence, or how could energy play a role here? So instead of focusing on all 10,000 birds, we're just focusing at 1,000 species pairs, sister pairs. We now have the phylogeny of all birds. We can reliably identify which species are sisters, and we can take those as units, and we can then look at their sympatry. So where in the world do these 1,000 sister species pairs overlap? And what we find is that they tend to overlap much more in some places than others. And in fact, that pattern that we see there is very, very similar to the pattern of overall species richness. The problem here is it's really hard to get an evolutionary handle at this pattern, because it's these wide-ranging species that are driving everything. We're unable to easily get at the phylogeny that's underneath all of that, right? Now, with this pattern, we actually have a very, very good handle on what's going on here. These are sister species. We know that through, for example, allopatry, they would have separated at some point, and then, in some cases, come back together again. So these are the places where the sister species exhibit sympatry.
Have you normalized it by species richness? What do you got?
That's
30?
that's
Yeah.
just the percent of pairs.
Oh, it's a percent.
Percent of
Okay,
pairs.
it's
Sorry, I should have
normalized.
been clear about that. This is percent of pairs. So the percent of pairs within a quadrat that exhibit coexistence or symmetry, secondary symmetry. Now, let's relate that to productivity, and let's not do it in a purely correlative way, because now we actually can get at it in an evolutionary way. We can actually model how the process of coming into symmetry or retaining symmetry in an evolutionary way in relation to productivity. And we can bring something else in, of course, the median species age, sister species age. So, of course, sisters that separated much longer ago have a greater probability of coming back into symmetry. Whereas if you only separated very recently, then chances are you haven't had the time yet to come back into symmetry. So you need to take that into account, and there is actually a clear pattern in the median age of these sister species. So we have one model that's a pure age model, where in a low energy grid cell and a high energy grid cell, we expect the same slope, the same rate of coming back into symmetry. Okay, and this would be a younger sister species pair, and that's an older sister species pair. Or we can have an energy dependence going on, where the slopes between high energy, low energy grid cell are different. So here we have the high energy grid cell coming, or the species in that pairs that are characterized by that grid cell or by that environment, coming back into coexistence again much more rapidly. So we test this model we run a Markov chain model of species coexistence, where we quantify these two parameters sort of coming out of coexistence or coming into coexistence, and test whether a pure age model does a better job or whether this energy dependent model does a better job. And we find that indeed, in addition to age affecting the propensity of coexistence, net primary productivity has a very, very distinct effect. We find that in high energy regions, this propensity of coming into coexistence and then not losing it is much higher than in low energy regions. And overall, we find that then productivity has a very distinct positive effect on coexistence incidence. And that is robust to a variety of assumptions about how you measure these things. And it is also the most consistent effect compared to other potential predictors, including, by the way, here the hand-wing index, which could tell you a little bit about dispersal ability, and also some other measurements of species that we've thrown into the mix here. Age of the sister species pair and productivity are consistently the strongest predictors. So we find that with this scale-conscious and hierarchical perspective, I feel we are able to do a better job in actually addressing the processes behind the spatial variation and richness, that this region view can be quite beneficial in getting a first understanding of diversification rates and how they actually don't matter in explaining this among regional variation. We find that at the assemblage at the grid cell level, energy availability is a key facilitator of coexistence, and that we have now new tools with these phylogenies at the sister species level, for example, to address this from a macroevolutionary perspective. And going forward, I think out of this I take the conclusion that we really need a better understanding of biotic constraints on coexistence over broader scales, so that we really need to take it to that fine scale, but over larger extents, and that we need to extend these sort of approaches to other species groups to generalize some of these ideas. And with the few minutes left, I just want to show a few ideas and next steps here that we're taking to go into this direction, that really try and help advancing the trait data, advancing the phylogenetic information, and that we need finer grain, larger extent spatial information. So these are the two projects that actually Michelle already mentioned in the beginning. And I really feel, yes, we can do with better theory. There's more to be done here, also across scales. We need more paleo data, of course. There's a variety of empirical information that would love to have to do a better job here. But I do feel that we certainly would benefit from this arena here, a more extensive trait data that would allow us to assess this trait or niche overlap that I mentioned earlier in a more rigorous way. So this was just the first attempt. And more complete phylogenies, uncertainty assessed phylogenies that would allow us to do more of these sorts of assessment that we just started to begin here. But let me first talk about the spatial data. I feel for synthesizing across these spatial scales, we really have been limited by the fact that at large extents we tend to, in macroecology, work with this really coarse grain data that gives us the large extent but doesn't give us these processes down here. In community ecology, we tend to work with data that's not spatially very large in extent usually, just in a country, often, and or has very, very large holes in it or is not representative. And I feel to achieve the synthesis, we really need more large extent, fine grain data. And that's, for example, where I think remote sensing can play a really important role. I talked about that last Friday in terms of policy, so better data for policy, for conservation. But I feel it's really also absolutely instrumental for advancing some of the ideas in this more basic field that I'm talking about today. And I think to get there, to get to that finer scale understanding of species distributions supported with remote sensing, we need better informatics infrastructure. Here's some stuff that we're doing in Map of Life right now to annotate different types of species distribution data with environmental information at increasingly fine scales. You really got to build infrastructure to be able to deal with this big data that's coming from remote sensing. We're able to then start to annotate even single movement paths here for, there's about 120 individuals of storks with data at 1 hour to daily resolution from 30 different populations. And it's amazing what you can do now in terms of trying to capture not just species and their environmental associations across the range, but also understand the granularity within the range, even down to populations and single individuals. On the remote sensing front, there's some real opportunities to come to an environmental characterization that's ecologically more valid. Here work by Adam Wilson, a former postdoc in our group, on cloud cover elevation globally, that's allowing us to capture the fine grain variation in environmental conditions at really amazing detail and allow us to then bring some of the species distribution predictions down to a much more reliable level as we're using that information rather than just WorldClim to get to really detailed fine-scale predictions of species occurrence and ultimately species richness. So I think that's the sort of stuff that would allow us to go to a finer spatial resolution, yet at contiguous continental to global scale, to then really test some of the scale-dependent ideas of the determinants of species richness. We're starting to integrate different data types in Map of Life, and let me just flash here in the background, encourage you to have a play around Map of Life. Here, for example, a solitary tinamou, you see different data types informing that range. They all tell a slightly different story. We need to bring different data types together. Have a play here. We're working with AmphibiaWeb to then bring that information, for example, back to the places where much of the real information is coming together for specific taxons. So you could place this in a different website. In the BirdLife project, we had a meeting here just a month ago or so, and Rory and Dave and Michelle here are some of the key drivers of the project. We're really trying to bring together different facets of biodiversity for that group, terrestrial vertebrates, and try to advocate that ultimately, yes, we could potentially look at terrestrial vertebrates as a model system for doing some of that stuff that I mentioned earlier, bringing trait and phylogenetic information together and doing some of that cross-scale inference that I mentioned earlier. Starting to have phylogenies for all of these groups, are able to assess the similarity and differences in this variation in diversification among them. And with that preview, I want to leave things, and thank you for your attention.
Cracked up to be the lineages jumping back and forth across the boundaries. Would the predictions fundamentally change? How critical is it to actually the underlying theory of how diversity might develop at this scale?
So let me take a question as one that's sort of just questioning how smart bioregions are as units, right? So in a way, that's what you're referring.
Well, I'm almost thinking let's let them be units, but but what if lineages were jumping across them? I mean, what if we took them at face value but allowed the lineages to jump? And then the next question is whether we should take them at face value.
Yeah. So I think we should. We can take them at face value to a certain degree, right? So we've done simple assessments of how much families or genera are restricted to them, and I remember actually Paul had to address that question also, right? So how useful are they? We find amazing conservatism, if you will, bioregion conservatism in these groups. Only in birds we find that whatever, I can't remember, 500, 800 species are really jumping across these bioregions, and then you have migratory birds, of course. Now, I think though it will be really interesting to address how clades have ultimately jumped from one bioregions to others. There is a postdoc in our group, Chris Trissos, who is actually trying to address that very question, so biome switching, right? And it's certainly going on, and it will certainly be really, really interesting question to drill deep there. I think this is just a starting point. Ultimately, I think we want to have spatial macroevolutionary models that allow that sort of thing, right? And Jesse, that sort of framework, Bissey, Jesse, where you have the state dependence, where you can actually bring in how much, you can actually account for that sort of switching, I think is ultimately a way forward here, but they're just still computationally a bit restricted to be able to do that sort of thing. So I think I really just look at that as a starting point, and ultimately we'd want to model multivariate environmental niche evolution along the phylogenies together with some of these overall rates of diversification and how then species of clades are sitting in certain biomes or not. So, but I think they are a useful starting point, and they certainly allowed us to get started with this thinking. Fair enough, Paul?
What are some of your ideas about how productivity promotes coexistence? I remember one idea being that highly productive areas may have, I don't know, greater habitat complexity or structure. What are some of the other hypotheses?
No, absolutely. So this is still phenomenological in a sense, right? We now know, okay, there seems to be a signal, but what exactly it is in detail is still an open question, right? So you could argue that ultimately this greater amount of food fluxing through the environment or energy fluxing through the environment would allow just species to coexist more readily. Because we're talking consumers here, it's actually the habitat structure, the vegetation that also may be affected by productivity and ultimately facilitating coexistence, right? So we're not able to separate that. We may still have a strong effect here simply of higher places with greater net primary productivity or actual evapotranspiration being places where vegetation was able to get more complex and thereby offering up greater habitat niches facilitating species coexistence. So those would be like the two most opposing views, and there are some that are in between. I think that as we get to better, through remote sensing, better capture of some of that heterogeneity in 2 dimensions and 3 dimensions, I think we'll increasingly be able to address that potentially. But it will still be a very, very tricky question.
Yeah. Go ahead.
Okay, so you've been thinking about time on long scales. What about annual scales? One thing that didn't come into play is seasonality. And the tropical ecosystems vary tremendously. So if you start thinking about latitudinal variation without kind of incorporating those kinds of seasonal effects, how do you play with that component?
Just trying to pull up here. So we did have seasonality. So statistically, just a purely statistical way. Yeah, I mean, it's kind of kind of this typical macroecological approach. Yeah, you throw in whichever way you happen to capture seasonality, and then you check, okay, does it explain as much or more, and it tends not to, right? So insofar, those would be the statistical attempts of doing it, and it can actually be quite important in greater the seasonality here. Actually, the more depressed coexistence tends to be, interestingly. I feel there's more to be done in birds and moving species, mobile organisms. You actually have a whole suite of challenges you need to address here. But in terms of just as a predictor of today's variation in species richness, yes, seasonality seems to have a statistical effect, but we haven't gotten to good grips yet of how it matters really.
So well, two kind of related questions. The one is, just to make sure I understood correctly. So in like South America, you were showing when you were doing the new trait stuff that you've been doing, that you're adding on average species that are more similar, or new species.
Yes. Now,
So is that a consequence of microallopatry because of the scale that you're doing it at? I mean, intuitively, you'd almost expect more species to be occupying slightly different sets of traits rather than being more similar. So is it a spatial element?
no, I don't think it's a spatial element, because
Or
you
just
find it
variables
at the
you
fine
can't
scale
fit.
also. What's going on here is a number of factors, right? So yes, as you look at places that have greater species richness, yes, and you add in more and more species, they tend to add in more and more traits and trait combinations, and your hypervolume of trait diversity tends to increase. But the question we are asking is, does it on a per species basis? And actually on a per species basis, as you verify that hypervolume, if you will, down to a standard set of species for every region, it turns out that places like the wet tropics in South America, when you measure birds with these sorts of, with the traits we did, they tend to look very, very similar. And as you add on more and more species, they tend to be more and more similar to the ones that are already there, as you sort of do that in a statistical sense. So what that does not account for, though, is potentially really detailed behavioral differences that obviously exist. Yeah. So I really consider this as just the first step and kind of an idea. Hey, we could perhaps get to grips with this in this way.
So related to that, does that support Rickliff's idea of communities not being saturated?
It's in that context, I would argue yes. Yeah. In that context, yes. I don't feel so this is ultimately getting at competition, right? So are we at the regional scale finding a signal of competition that would impede speciation, right? That's kind of what that is asking. As you get, because you filled the space, right? But we find that in South America, these ovenbirds, these tyrannid flycatchers, they were pumping out species, and they tended to be very, very similar. And we're talking about this at the bioregion scale, because we felt like it's at that scale that ultimately, over evolutionary timescales, things could have interacted with one another. Yeah? And I don't believe there is a signal of competition, right? Actually, at that scale. And in invertebrates, or in birds for sure, I've yet to see some really good demonstrations, even at finer spatial grain, of this competition. Yeah?
Can I have one more question?
Yeah.
So when you calculated the mean age of your sister taxa,
Yeah.
it's really striking what you see for like Africa and Indonesia.
Yeah.
Do you think that could be a consequence of the taxonomy of those regions being so understudied relative to the New World? Like it could be a data artifact?
Yeah, I know you already want to use that figure for your next grant. So if NSF is listening, then I should say yes here. It would have to be quite dramatic. It would have to be quite dramatic, and we can get into the details here. And you'd want to do some sort of quantitative analysis, but sort of shooting from the hip right now. Call up that map again real quick. It would have to indeed be quite dramatic to lead to these sorts of differences. But I still feel there's lots of work to be done in Africa.
So among people who study the genetics of speciation, there's been a real interest in the last 5 or 10 years in ecological speciation, right? And so, but you're finding in the bioregions, the sort of time area aggregated is explaining most of the species richness, and that there are a lot, for the traits you measured, you're not seeing big differences. Does that argue against ecological speciation?
I feel it's hard to pin that down with the data we have at the scales that we have it. So we very much think in the case of birds about allopatric speciation. That's how we usually approach it at these scales. I feel I wouldn't want to venture a direct yes/no response on that one. I think there is still a lot to be understood, but this may not be the right system or the right sort of data to do that about how much ecological speciation may matter.
I guess more generally, as somebody who studies speciation, I always feel like there's this almost philosophical divide between people who are studying large-scale patterns of diversification like this and people who are working on particular examples and trying to get at mechanisms. Do you see ways of bridging that with the kinds of studies that you're doing?
By actually talking more with one another and drill a little deeper there, right? But I think by bringing it back down again to that sort of stuff, right? To really get the data at increasingly fine spatial and ecological scales ultimately down here, right? The problem is to really get at ecological, we need it over longer time scales also, that we usually don't easily get when we talk about species and species of logics, because we're talking about millions of years, not just a few thousand or tens of thousands of years, right? So it's really tricky. I think the more I do feel that ultimately this field of landscape genetics, phylogeography, right? There are real opportunities, I feel, to connect these fields up all the way to the below species level, and it's at that level that I think we can get at some of these factors and separate out environmental associations, barriers, and issues, and then factors such as ecological sort of separation in situ or not. So I feel addressing connecting up the scales would be my response. Yeah, but also scales in terms of organismal organization, not just spatial scales. So going below the species level.